Hey there, I'm Shelby,
Hey, I'm Megan.
and this is the rest of the story.
Today, we're going to be talking about the neuromuscular junction and
Oubain. All right, I'm going to start this off.
All right, Shelby, take it away.
So, the neuromuscular junction starts with the action potential, which you can see right here at the axon hillock membrane. So, on the outside of the cell, we have a high concentration of sodium ions. On the inside of the cell, we have a high concentration of potassium ions. Right here, you can see right now the membrane's at resting membrane potential at negative 70 millivolts. So on the inside of the cell is negative. And you may ask why, and that is because proteins make the inside the cell negative despite potassium ions being plus. And then outside the cell is positive because there's a high concentration of sodium ions that make that positive. All right, so over here we have a ligand-gated sodium ion channel, and when a ligand attaches to it, it catalyzes the opening of the gate, allowing a rush of sodium ions to flow through into the cell membrane. So, through active transport, sodium ions flow into the inside of the cell. So this little bit of positive charge then catalyzes the voltage-gated potassium ion channel to open. Potassium ions flow to the outside of the cell, also using facilitated diffusion. At this point, the cell has depolarized. It is happy, and these ions have moved with the concentration gradient. The action potential then continues to move down the membrane, and then Megan will talk about later how that affects what she will be talking about. All right. Then, to restore resting membrane potential, which is negative 70 millivolts, the sodium-potassium pump needs a little bit of ATP to get to work. The sodium-potassium pump uses active transport to push sodium ions and potassium ions across the against the concentration gradient. So it will take three sodium ions and push them back out to the outside of the cell. So these three will reappear here. And this is against the concentration gradient again. So this is making it resting membrane potential again. Oops. And then it takes two potassium ions and pumps them back out to the inside of the cell. This restores resting membrane potential, allowing the neuron to continue to send action potentials again and again. All right, Megan, you can take it away.
All right, so what might be happening after this action potential is passed down the axon, you ask? Well, it reaches the terminal buttons of the neuron, where the neuron connects with a postsynaptic cell, which in our case is a motor neuron connecting to a muscle tissue or other cell in the body. So this connection here is called the neuromuscular junction or the synapse. So in the synapse, the terminal button first receives the action potential. So these are voltage-gated sodium ion channels, which receive the high voltage from the nerve impulse and open, letting sodium ions flood into the cell. Once the sodium ions flood in, the voltage increases and causes calcium channels to open and flood calcium ions into the cell. Once these ions are inside of the cell, they trigger vesicles containing acetylcholine molecules to merge with the presynaptic cell membrane and release the acetylcholine molecules into the synaptic cleft. Here, they travel to the postsynaptic membrane and bind to acetylcholine receptors, which are ligand-gated cation channels. So, as you can see from this zoomed-in picture, the acetylcholine molecules are binding to the receptor, which before they bind is closed. Then, once they are attached, the gate opens and allows sodium ions to flood into the cell and potassium ions to flood out of the cell. So, this is similar to what we saw here. It's just starting with acetylcholine as the ligand. And when this happens, the action potential travels down the membrane and starts the action potential in the next cell. So, after this happens, what happens to the acetylcholine? Well, it is broken down by acetylcholine esterase, which is the enzyme that breaks acetylcholine down into acetic acid and choline. And these, if we refer back to our big diagram, break down and travel back into the neuron and go into a vesicle where they're combined again into acetylcholine and reused into the next action potential that is sent through the neuron. So, this finishes up our transmitting of the neural impulse and the sending of it to the next cell. So, what we're here to talk about today is mainly Oubain and what it does to animals. So, what is Oubain, Shelby?
Oubain is a toxin that was used by ancient African hunters. So they put it on arrows to kill animals effectively and quickly. And let's see how that looks with the sodium potassium pump, Megan.
So, Oubain was a toxin that attaches to the sodium potassium pump and binds to it to inhibit it from working properly. So, it will take the place of the sodium and potassium ions so that the sodium potassium pump can't pump sodium back outside of the cell and potassium back inside of the cell. And since the sodium potassium pump is constantly working as the action potential travels down the axon, it is necessary for the transmitting of a second signal to go down the neuron. So if the sodium potassium pump isn't working properly, the sodium ions will be stuck on the inside of the cell and the potassium ions on the outside of the cell. So the next signal can't go through and the next voltage-gated channels won't work properly.
Yeah. So basically, the cell will always be, or the membrane will always be depolarized and it'll never go back to its resting membrane potential. And if we were to predict what this would do to an animal, if it was, for instance, using like an arrow, it was dipped in this Oubain and the animal was stabbed or something like that, since it inhibits the sodium potassium pump, which is necessary for all of our neuron functions, basically the next time it was stimulated by a ligand or anything, the animal would drop dead because it would no longer be able to function and move. So there's that.
Mm-hmm.
This was the rest of the story featuring Oubain.
Thank you very much.
Thank you for watching.
See you later.
See you on the flip side.